So yeah, let's jump right in. Thanks for the opportunity to present this update from the Genotox Technical Working Group. And of course, first and foremost, I want to really acknowledge this excellent group of experts that are really joining Christine with my co-chair and I really doing all of this work with a lot of very valuable input from the team. So much appreciated. And of course, also Connie, invaluable team member here too, that keeps us straight. So now, what are we doing? And I think you'll probably see that kind of working group mission in a similar form from the other teams too. We are trying to develop a screening strategy that can reliably identify potential genotoxic botanicals with future application to evaluate associated human health risks. And so, I mean, a few considerations here to get started. I mean, there is quite a lot of established in vitro methods in the area of genetic toxicology, which is not the case for every endpoint. But I want to mention here, and that's important to me, and I'm not contradicting you, Cynthia, from what you had said in your initial introduction, but I think it is important to realize that sensitivity of an assay is important, of course, very, very much. But of course, overall predictivity is also important because genetic toxicology is an area where, because these methods have been really developed quite a while ago, some of them, like the Ames test 60 years ago, the focus really was on pushing them towards very, very high sensitivity at the expense of specificity. When you start putting them back in together in test batteries, you really have a super sensitive approach, but you might not be doing very well anymore on the side of specificity. So you'll be generating a lot of positives in the context of something that's not relevant for the human situation. So I think you need to have the right balance, but of course you need to have very, very sensitive battery here. So we are focusing on screening level assays. In silico modeling was the first really that we approached, followed by in vitro guideline studies, and then also some of them more novel follow-up tests, and I'll explain that strategy. Actually, the Genotox team has nominated 7 botanicals, but we are, and I was going to say we are evaluating all 13, but of course from the latest slides I've seen that now there's 16 on the list. But I mean, so far in our program, we have 13 extracts that we are evaluating. These are botanicals with existing data, and we selected them as case studies. But I want to emphasize, not for all of those, because some of the nominations came from other teams. We do have a firm grip of what the expected in vivo outcome in Genotox would be. So the 7 that we've nominated, we think we have a pretty good grip on that. And so I'll focus on examples from that. Now, for the in silico work, chemical analysis, as was explained earlier, allows for the authentication and constituent ID and quantification. But I mean, what I'm presenting here represents the status 2 years ago. So the analytical program wasn't finished at the time. So when we did the in silico, we had a lot of phytochemicals in there, but I think we probably have to redo it with what's on the list today. If it's worth it, I mean, I'll talk about that in a couple of minutes. And then, of course, at the end of the day, we want to develop a toolkit and a framework that's really basing on the data that we have collected by then. So the botanical nominees are on the list here. Those are the ones that our team really nominated. And of course, it's led by Arceloria Fangchi, which, I mean, contains azelaic acids. And of course, I made that comment earlier, really one of the very potent mutagenic plant-based mutagens we know. And I mean, even there, and that's the other point I wanted to highlight, because that's one of the differences, I think, compared to some other teams. I mean, it's not so easy to link a health effect in humans with a cause, right? If it's something like development of cancer and nobody screens for mutations, at least not today, how they're piling up in humans. So if somebody develops a certain cancer, it's almost impossible, or up to now it's changing, to kind of link it with the cause. So I mean, history of human use or safe use is kind of limited in this space. And even for Arstelochia, it took quite a long time, and actually some kind of unfortunate events that happened that really allowed for unraveling this story. But I mean, that's one of our leading positive candidate here. And then, of course, you'll probably see some other familiar names. We have others in there, like, for example, green tea, and I'll talk about that a little more too, because I mean, it's something that everybody is familiar with. But what you can see here, and in the column of existing experimental results, it is not a clean negative necessarily in all the Genotox in vitro assays. But I'll get there again. Now that's how our workflow, the strategy the team agreed on really looks like. I mean, basically starting with in silico prediction models, and we had done an initial analysis that I'll briefly present. This was done with the lead scope, what's called the Genotoxicity suite, which is a combination of certain assays. The results are predicted for AIMS and other assays, including even micronucleus in vivo. But the strengths of these modules is not equal, right? There's a lot of data in the AIMS module. There's much less data in some of the other modules. So kind of take it with a grain of salt, what you'll see in a couple minutes. Then, I mean, the idea was to have a tier 1A, which is our initial screening battery, which we decided to be AIMS, the classical AIMS, guideline AIMS, and in vitro micronucleus, because that's a battery that captures all what we call the genotoxicity endpoints, gene mutation, blastogenicity, and amogenicity. And if both are negative, such an extract would be deprioritized for further testing. That's at least the idea. And then we have a tier 1B to do follow-up, because, like, for example, for gene mutation with a mammalian cell assay, because certain classes of chemicals are not predicted very well by AIMS and so on. And I mean, there's other ways to do other follow-up. And then we have follow-up tools listed kind of in a bucket, so to speak, that can be used to gain mechanistic mode of action information, or, for example, some higher tier in vitro assays like a 3D skin model, liver spheroids, potentially HENSEC micronucleus COMET assays, but also DNA damage response assays. And I mean, we have, since my lab is really doing a bit of ToxTracker work, and we're involved in the international validation efforts, we volunteered to kind of generate data with that assay. And I think we were able to fit this in rather quickly. So that's so far the only finished data set. But we do have data on the AIMS, and I'll be able to make some comparisons here in a bit. Now, and I don't see any comments or raised hands and so on. If something is really unclear to you, and please feel free to interrupt me. So now we have the initial screening results, and this is based on 246 constituents from 13 botanicals, and that's again the lead scope suite. And the botanicals didn't all have the same number of ingredients, so that ranges from Arstelochia only 3, and comfrey with 40 identified constituents. But what you can see is that the red bar basically represents positive results in a certain number of constituents. And you see already there's a lot of red in there. Now, when you look at it kind of again in a test battery approach, actually 73% of all the constituents from these plants were positive in at least one model. So I mean, it's really probably not going to be a great model for us to distinguish genotoxic from non-genotoxic extracts, and that's the complexity of of what we're dealing with, this plant phytochemistry is also quite complex and limited good data represented in these initial in silico screening models. So that doesn't seem to have gotten us very far. I just wanted to highlight green tea again because I brought that up. Actually, the in silico modeling predicts it as positive for glastogenicity and for gene mutation in mammalian cells, for negative in bacteria, and actually for positive in the rodent micronucleus assay. So the actual data set from NTP with another batch than what we had been looking at is almost exactly the opposite, right? It was positive for IMS, negative for mammalian cell, and it was also negative in vivo. So again, not looking that great. Now the ToxTracker assay, and I'm limiting it to 5 compounds because there's not enough time to go through all of them. It's a reporter assay that's really using mouse embryonic stem cells, and it uses reporters that have been found to be quite predictive of certain outcomes. And it doesn't only look at DNA damage, it also looks at other endpoints. It looks at oxidative stress, it looks at P53 activation and protein damage. Has shown quite nice sensitivity and specificity. The validation paper is in the works. So that's something we really I think, using more and more also in our lab in Procter & Gamble. The work was done by Ashley Alaman. Very excellent support here. Now my slide isn't switching. Here you go. And I think at this point, it may not be a big surprise to you that's Aristolochia franchi. And if you want to focus on maybe the blue and the red, right? The two red one are the two gene tox markers. What you see, the dotted line here on these left-hand graphs that shows the induction. So 3 means threefold. Actually, the cutoff is a twofold, so a doubling of is considered a clear positive. So you can see it goes up very high, and it goes up even at levels where it's not that cytotoxic. And actually, in the plus S9 means with rat liver homogenate, which is a standard metabolizing system in Genotox study. Minus means without it. So you can see that even already at 10 microgram per milliliter, you see a clear positive for aristolochic acid. And actually, the sample we are testing, we know now, has 1% of it in there. So I think it's fair to assume that even if there was much less aristolochic acid in there, we could have detected that as positive in this assay. And it's also an assay that, for example, could be used for screening purposes or for potency evaluation. Now, comfrey extract, that comfrey extract contains PAs, but it doesn't contain actually a high amount. I don't have that one in my head, what exactly it was, but it's a much lower content. And what we see a positive response. We see a pretty strong oxidative stress response, but we see that the markers exceed a twofold. So this will be a positive ToxTracker result for comfrey. Now, golden seal, again, it shows a fairly clear positive response, both with and without S9. The gray shaded area means that it exceeds the cytotox cutoff of 75%. So that data point basically wouldn't count, right? Just to explain that. So you don't typically look at that. I left it in there because I think it's kind of interesting also in the context of green tea, which is the next after this. Ginseng, an expected negative. So all the first 3 were expected positives. They all tested positive. Comfrey, a little more borderline than the other 2. Ginseng, expected negative, shows a very, very clear oxidative stress response, but no response in the DNA damage markers. Green tea actually is also overall considered negative. But I mean, you can see fairly clearly, again, that the last data point doesn't count, But even with a data point below, you're getting a bit closer to the twofold cutoff. So there's clearly something happening here in the background for green tea extract. So it's a negative that you will see in that next table, which compares the ToxTracker result and Ames result for these 5 compounds. I put in brackets. Now, the Ames results are results from NTP testing. Data was generated, I believe, at ILS, and I think now different name. The Ames data, to make sure that's clear, are preliminary data. They need confirmation, which is ongoing. But I mean, you can see that the results line up actually, to me, almost surprisingly well between Ames and the ToxTracker. Istolochia and Golden Seal, clear positive. Comfrey was a positive in ToxTracker, but I mean, just exceeded the threshold of 2. In Ames, equivocal, but that may change with the repeat. Green tea also showed something in both assays, but didn't trigger the threshold. Sorry, I didn't mean to switch. And ginseng was a clear negative in both assays. So I think we have good alignment. Obviously, the green tea extract showed increases, and we really, I mean, there was also something. The batch we are using for green tea, it's a different batch than what was tested in NTP and led to a positive response. In vitro micronucleus assays are underway. So with that, just quickly, next steps for us, of course, first of all, is finish all that stage 1A testing, so Ames and micronucleus, and then initiate 1B tests, stage 1B test. Then, of course, also potentially do more mode of action studies, but we want to have the full data set in our hand before we make any decisions. And then, I mean, again, at the end, we want to evaluate the performance of that test battery and potentially usefulness of follow-up tools for all these nominated substances. Of course, also at the end, publish and communicate the results. So I think that brings me to the end, again, acknowledging all the great work of the team and happy to answer any question you may have.
That was really great, Stefan. Thank you for walking us through that. There are a few questions so far. So a few from Andre. He asked about cell types. So could you comment on the reason of using MECS instead of human? So I guess that's a mouse cell line. I don't know if you can see the chat.
I just open it now.
Okay.
I mean, I can. This is really what that's the company that has developed an assay, Toxis. Really, that's the model they brought forward. I think they have an assay, or have tried an assay basing on human stem cells. But I mean, this assay works very, very well in their hands. Also, that's what the validation seems to show. So while it may not be intuitive that it's mouse-based stem cells, it does work. So that's what we have in our hands right now.
Then he had a follow-up asking if there's significant differences in DNA repair and chromosome stability between mouse and human cells.
Well, I mean, again, I think these cells, as far as we know, I mean, in terms of DNA repair, I mean, contrary to some of the cell lines that are used in standard testing, are P53 competent, right? And they also DNA repair competent. I'm not sure there have been any direct comparisons done in that space.
Okay, there's a question in the Q&A from Kaylee asking about the results in the summary table comparing green tea and milk thistle. It seems that green tea had more positive data, but the expected outcome is non-genotoxic compared to the less positive data in milk thistle, which is predicted outcome of genotoxic. Do you have additional information on this?
I wasn't sure I fully got it. Let me
paste it. Sorry, I think only I can see the Q&A. So if people do things, please put it in the chat. And I don't know if we mentioned this, part of the green tea we selected was a very concentrated extract that was also decaffeinated. So it's not, just the green tea you drink.
Yeah.
So that might explain some of the results. But I
Yeah.
think in some of the NTP, their bioassay, it was negative. So that's why we're expecting an in vivo negative result. Is that correct, Stefan?
Yes, that is correct. Thank you. And then I realize as we talk about that, I think I put the wrong name on top of the comparison table in terms of the extract for that fifth extract. But yeah, I can correct that later. And thanks for the hint that this is unpublished data, so please do not share.
Yeah. And oops, sorry. I think there's one other question. Oh, Jim he put it in the main chat. Just the point about the Aristolochia poisoning that happened in Belgium.
Yeah. Appreciate that, Jim.
Yeah. And then Dan Roberts had a question. Could some of the antioxidant responses driven by NRF2/4 be on target effects on the component of the extract?
Sure. I mean, it's always. I mean, that's of course why we think we have a fairly high percentage of positive testing results with in vitro assays in Genotox because it contains a high. For example, a lot of them contain a high percentage of flavonoids, right? And of course the oxidative stress pro-oxidant versus antioxidant is a bit a complicated story. So I think most antioxidants are pro-oxidants at high doses, which is what we use in Genotox assays.
Time. Oh, that was just an explanation. The only other question I see so far is from Corrado asking about our cell type for the micronucleus. And I don't think it's not HEPA-RG. Is that correct?
No, not yet.
It's the
That was a
TK6.
potential, but TK6, human lymphocytes, TK6 cells.
Yeah.
Is what the screening is being done. But we want to potentially look at other, also HEPA-RG, for example, in depending on the results.
Great. Well, thanks for that, Stefan. That was a really excellent overview. I don't see any more questions. I think we can then turn over to the rest of our presentations today.